Hello everyone, and today we'll be discussing the NCERT exercise for the chapter Biological Classification. So before starting this lecture, I just want to give you one information that the images, text and other materials used in the presentations are for education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So, what we will study and what are the objectives of this lecture? So, we'll discuss the NCERT exercise and we will understand the concepts which are related to the chapter biological classification. So, these questions they are based on some concepts. So, we will understand these concepts as we are discussing the NCERT exercise. Okay, so we'll be discussing the NCERT exercise for the chapter biological classification. So what is the first question here? Discuss how classification systems have undergone several changes over the period of time. Clear? So classification system has changed a lot. There is refinement of the classification system. Okay. So let's discuss the answer. So the evolution of the classification system is a very fascinating journey in the field of biology. And this reflects what? This reflects that our understanding of the organisms, of the living organism is increasing. We are now able to better understand the living organisms. We are able to understand the evolutionary relationship between these organisms. Okay. Now we are studying this organism at molecular and genetic basis. We are also studying the evolutionary relationship between these organisms. Means we are able to better understand these organisms. And also the techniques, the microscopic techniques, or microscopy, it is better now. Then new, you can say, living organisms are being discovered. We are now able to reach the remote areas, and new organisms they are being added. And the previously known organisms, we are also able to better understand the previously known organism as well. So this is leading to the change in the classification system. Okay, this is clear. Now here's a broad overview of the key stages in the evolution of the kingdom classification. So early classification system, let's talk about the early classification system. So these classification system, they were based on the appearance and the behavior as well as the utility to the humans. So it didn't have any scientific basis. They were just based on the appearance of the organism, their behavior, and how are they useful to the humans, how much useful they are to the humans. Then came the classification of Aristotle, who divided the living organism into two groups, plants and animals. So this classification given by Aristotle was primarily based on the mode of nutrition. Okay, remember this. Primarily, it was based on mode of nutrition, whether the organism is autotrophic or heterotrophic. In addition to this, he also considered some characteristics, some morphological characteristic as well. Okay, so he divided the plants further into herb, shrub, and trees depending on their size. Then the animals, they were divided into two groups. One group of animals had the red blood cells, means red blood. Other group of animals, they didn't consist of the red blood or the red blood cells. So this had little bit of scientific basis. Then came the Linnean system of classification. So Linnaeus, he was a Swedish botanist. Okay, he proposed a hierarchical system of classification that formed the foundation of the modern taxonomy. So again, he gave two kingdoms, kingdom Plantae and kingdom Animalia. Kingdom Plantae includes all the plants, kingdom Animalia includes all the animals. And his classification was based on overall structure and reproductive characteristic also. So he also included reproductive characteristic, okay, into his classification. Now, clear? But there were some limitations to the linear system of classification, that the unicellular and multicellular organism were placed together. Okay, autotrophic and heterotrophic organism were placed together. Unicellular, multicellular organism were placed together. Prokaryotic, eukaryotic cells were placed together. So again, there was many organisms that didn't find any place in these two kingdoms. They were not fitting in these two kingdoms. So there was the expansion of kingdoms. Okay, more kingdoms were added, like kingdom Monera, kingdom Protista, and then kingdom Fungi. Okay. Now, as the scientific knowledge expanded, means we were now able to better understand the living organisms by using new techniques, okay, by studying them at molecular and genetic level, by studying the evolutionary relationship, we are now able to better understand the living organisms. So it became evident that two kingdom system was inadequate to represent the full diversity of the life. Means lot of the organisms didn't fit in these two categories, kingdom Plantae and kingdom Animalia. So in late 19th and 20th centuries, additional kingdoms were proposed, such as kingdom Protista, which includes all the unicellular eukaryotes, and kingdom Monera, which includes prokaryotes. Okay. Then later one more kingdom, kingdom Fungi, was also added. Now then came the five kingdom system given by Whittaker. Okay, in 1960s and 1970s, biologist Robert Whittaker proposed a five kingdom system which aimed to better reflect evolutionary relationship and cellular organization. So Whittaker's five kingdom classification, it also represented the evolutionary relationship. Okay, it also represented the cellular organization. What do you mean by cellular organization? Means they are unicellular or they are multicellular. Okay, or what else? And what else? Whether they are made up of cells, tissues, organ and organ system. So one was cellular organization. Okay, organization of the cells, how the cells are organized. So these were, you can say, the five kingdom classification reflected the evolution relationship also, as well as the cellular organization. Okay, because the Monera include all the prokaryotes. Then from Protista, all the organisms are eukaryote. In Protista, in fungi, Plantae and Animalia, all these are eukaryotes. Only Monerans, they are prokaryotes. Okay, clear. Then Monerans, okay, members of kingdom Monera, they are unicellular prokaryotes. Protist, they are unicellular. Monerans, they are unicellular prokaryotes. Protista, they are unicellular eukaryotes. And fungi, they are multicellular eukaryotic and eukaryote. Okay, there is only one exception, that of yeast, which is unicellular. Then kingdom Plantae, they are eukaryotes. Animalia, they are also eukaryotes. So five kingdoms were Monera, which includes bacteria, Protista, which includes single cell eukaryote, fungi, which includes obviously fungi, which are mostly multicellular except yeast. Plantae includes plants and Animalia includes animals. So this system highlighted the evolutionary divergence of many groups and provided a more comprehensive framework for the classification. Okay, so this five kingdom classification demonstrated the evolutionary relationship, or you can say evolutionary divergent, how the one group is You can say related to other group, or how much one group is different from the another group. You can say in both ways. Okay, so it reflected the evolutionary relationship, you can say, or you can say it highlight the evolutionary divergence also. Now, then there was a 3-domain system. This was given by Carl Weiss. Now, due to advancement in molecular biology, particularly in the use of ribosomal RNA sequencing, this revolutionized our understanding of the evolutionary relationship. So now we were able to better understand the evolutionary relationship. Okay, because now we were able to do the ribosomal RNA sequencing. So by this ribosomal RNA sequencing, we can tell about the evolutionary relationship. Okay, so then came the 3-domain system. What were the 3 domains? So they were archaea, bacteria, and eukarya. These were the 3 domains. Archaea and bacteria, they include the prokaryotes, and the eukarya include the eukaryotes. The kingdom Monera was divided into 2 domains. So there were 3 domains: archaea, bacteria, and eukarya. Archaea includes all the primitive bacteria known as extremophils. Okay, so there were 3 domains: bacteria, archaea, and eukarya. Now, the SIS system emphasizes the fundamental division between prokaryotes and eukaryotes. Clear? Now, there is continued refinement. Now, due to better molecular techniques, due to modern molecular techniques such as DNA sequencing, by DNA sequencing, we can tell about the evolutionary relationship. Okay, clear? So, because of this, we are able to better understand the evolutionary relationship. This is the reason for the refinement of the classification system. Okay. So there will be continued refinement in the future also. As we are discovering new species and the techniques are getting better to characterize the living organisms, we are now able to better understand the living organisms. There are discovery of new organisms also. So because of this, there will be continued refinement in the future also. Now the taxonomics, they employ a combination of morphological, biochemical, and molecular data to reconstruct phylogenetic trees and refine the classification schemes. Now, the ongoing exploration of microbial diversity, particularly in extreme environments, has led to the discovery of novel taxa and the refinement of existing classification systems. The next question, state 2 economically important use of heterotrophic bacteria and archaebacteria. So let's have a look at the answer. Heterotrophic bacteria, first of all, they are used in bioremediation. So heterotrophic bacteria play a crucial role in bioremediation process. They can degrade various organic pollutants which are present in soil, water, and air. Examples are species of Pseudomonas and Bacillus. They are used in bioremediation of oil spills, contaminated soil, and water. Then these bacteria break down hydrocarbons and other toxic compounds into harmless byproducts, thus cleaning up the environmental pollution, reducing remediation costs. So first is bioremediation. Then food production. Certain heterotrophic bacteria are utilized in production of fermented foods and beverages. For example, lactobacillus, which is lactic acid bacteria, and streptococcus species are used in fermentation of dairy products such as yogurt, cheese, and kefir. So these bacteria convert or they produce lactic acid, which contributes to the characteristic flavor, texture, and preservation of these food products. Additionally, species like Saccharomyces cerevisiae, so this is yeast. Scientific name of yeast is Saccharomyces cerevisiae. They are used in the fermentation of bread, beer, and wine. So in yeast, what happens? Alcoholic fermentation, which is kind of anaerobic respiration. So yeast is known as baker yeast. It is used in bread making. Okay, it is also known as brewer yeast, used in making alcoholic beverages. So during alcoholic fermentation, alcohol as well as carbon dioxide is produced. So this yeast, it metabolizes the sugar to produce alcohol and carbon dioxide. The rise in dough during the bread making is due to release of the carbon dioxide. Now, biotechnology, archaebacteria. Let's talk about archaebacteria now. They are the primitive bacteria. They live in extreme conditions. So they can be used in biotechnology. So archaebacteria, particularly extremophils, they thrive in extreme environment. So they offer valuable enzymes and metabolic pathways that are used in biotechnological applications. Enzymes can be isolated from these extremophilic archaebacteria and they can be used in DNA replication. They can be used in polymerase chain reaction because these enzymes, they are thermostable enzymes. They do not degrade at higher temperature. They can be used in molecular biology, genetic engineering, and medical diagnosis. So example is Tag polymerase. This is an enzyme which is isolated from Thermus aquaticus. Thermus aquaticus live in hot springs, means it can survive at high temperature. So an enzyme tag polymerase is isolated from Thermus aquaticus. This tag polymerase is a kind of DNA polymerase which is used in PCR. By PCR, you can amplify the DNA. Okay, so during PCR, very high temperature around 95 degrees Celsius is used to denature the DNA. Okay, so at this high temperature, the enzymes which are proteins, they get denatured, but tag polymerase, that is thermostable, this doesn't get denatured at high temperature. Then bioenergy production, archaebacteria, particularly methanogenic archaebacteria, methanogens, they produce methane. So they are utilized in anaerobic digestion processes for the production of biogas. So methane is the major component of biogas. So methanogens can be used for the production of biogas, which is used as a biofuel. Okay, so methanogens, they convert organic matter such as agriculture waste, sewage sludge and organic residues into methane gas through anaerobic fermentation. Okay. So this biogas can be used as a renewable energy source for heating, electricity generation as well as vehicle fuel. So methane production by methogenic archaebacteria contributes to efficient recycling of organic waste also. Clear? And the reduction of greenhouse gas emission as well. Next question, what is the nature of cell wall in the diatoms? So let's have a look at the answer. So this question is from the section 2.2 of your NCERT book. So you can find the answer there as well. So what is the answer? So you know the cell wall of diatoms, it is composed of silica. Okay. So they have a very unique cell wall. Clear? This unique cell wall is known as frustule. Now frustule, it has two, you can say, parts, epitheca and hypotheca. Epitheca is the upper half, hypotheca is the lower half. Now here comes some key characteristic of diatoms. First is the silica composition. So diatoms, they are primarily composed of hydrated silica. And this silica is deposited as intricate patterns of silica dioxide. These silica structures are extremely durable and can withstand various environmental conditions as well. The next is ornate structure. So these silica frustules of diatom exhibit remarkable diversity in shape, size and ornamentation. So the frustules, the unique cell wall of the diatoms, okay, they exhibit, they show various shape, size and ornamentation, and this, you can say, is very specific. It is species specific. Clear? So what does this mean? They are species specific. So by looking at the pattern of this silica frustule, you can tell about that species of the diatom because it is species specific. Each species has its own pattern. Now two-part structure means the cell wall of diatom consists of two overlapping halves known as epitheca and hypotheca, which fit together like a box and give appearance of a soap box. Now two-part structure allows diatoms to regulate the cell size and shape while providing protection as well as support. So that's why the cell wall is in two parts. Now permeable to gases and nutrients. So this cell wall, which consists of silica, they are permeable to gases and nutrients despite having silica in them. So this permeability allows for the exchange of gases such as carbon dioxide and oxygen, as well as uptake of essential nutrients required for photosynthesis and growth. You know, diatoms, they can photosynthesize. They are the chief producer in the oceans. So through the cell wall, there is exchange of gases such as carbon dioxide and oxygen. And through the cell wall, they can uptake nutrients also. These nutrients are required for photosynthesis as well as for the growth of the diatoms. Biogenic silica formation. So diatom possess specialized cellular machinery for the synthesis and deposition of silica, involving silica transporter, silicic acid transporter and silicifying enzyme. So this process known as biogenic silica formation enables diatoms to construct their intricate silica frustules. Okay, so this is little bit of extra information, biogenic silica formation. So basically what is happening, diatom consists of a specialized machinery which helps in the deposition of the silica. Okay, clear? So this process is known as biogenic silica formation, and this helps the diatom to construct their silica frustules within the cell wall. Next question, find out what the term algal blooms and red tide signifies. This question is from the section 2.1 of your NCERT. You can find the answer there. Let's have a look at the answer now. What is algal bloom? Now what happens? Algal bloom refers to rapid increase in the population of the algae in the water body, which causes decoloration of the water. These blooms can occur in freshwater and marine environment. So what happens? Algal blooms, they are formed in the polluted waters or the waters which consist of high nutrient. Okay, high nutrients. They are rich in nutrients, which is known as eutrophication. So basically, due to the excess amount of nitrogen and phosphorus, the algal bloom occurs. There is rapid multiplication of the algae. This is algal bloom, and this may cause decoloration of the water also. So algal blooms typically occur when conditions such as high nutrient levels, which is known as eutrophication. Okay, this is the first condition for algal bloom: eutrophication, high nutrient level, warm temperatures, and calm waters. These all favor the rapid growth and reproduction of the algae. Okay. Algae which are involved in algal blooms, they belong to various species of algae, including cyanobacteria, which is blue-green algae, diatom, dinoflagellates, and green algae. So these algae can form the algal blooms. Now, not all algal blooms are harmful. Some species of algae produce toxins as well, and these are known as harmful algal blooms. So these toxins can be very dangerous for the aquatic life. So they are known as harmful algal blooms, HABs. Okay. Now what happens? There is the overgrowth of algae. Now this algae will die. Organic matter will accumulate in the water. Now for the decomposition of the organic matter, oxygen is required. Okay. So there will be less oxygen available for the aquatic animals like fishes. So these fishes can die. means there is an increased BOD for the fishes, increased biological oxygen demand for the aquatic animals like fishes, because oxygen is being used for the decomposition of the organic matter here. Okay. So we were talking about harmful algal blooms. So these toxins can be detrimental to the aquatic ecosystem, to the human health as well, and aquatic organisms. A common type of harmful algal blooms include microcystis, acid toxins, and domoic acid. Now let's talk about the red tide. So red tide is also a kind of algal bloom. Here also discoloration of the water takes place. Now what happens? There is a red dinoflagellate known as gonilocks. So there is a rapid, you can say, overgrowth of gonilocks. Clear? Or you can say due to rapid multiplication of gonilocks or due to overgrowth of gonilocks. So what happens? This gonilock, this secretes a red toxin, red color toxin, which makes the color of the sea red. And this toxin again can be very harmful to the aquatic animals. Clear? So this is known as red tide. Now the term red tide can be sometimes misleading, as not all red tide result in red color water. The water may appear red, brown, green, or even discolored without any visible hue change, means color change, depending on the species involved. So gonilocks usually secretes red color toxin, but sometimes the color can be brown, green, or even discolored depending on the species which is involved. Now red tides can be harmful if the dinoflagellate responsible for the bloom produce toxins. So like gonilocks, they produce toxins. These toxins may even accumulate in the shellfish and the filter feeding organisms, leading to seafood poisoning in the humans as well. So this toxin can reach to humans also, who consume the contaminated shellfish. And obviously these toxins are, you can say, cause harm to the aquatic animals also. In addition to toxin production, red tides can also cause ecological disruptions by depleting the oxygen levels in the water, resulting in killing of the fishes and have negative effects on the marine life. So again, oxygen will be utilized for the decomposition of the algae, and there will be less oxygen available for the aquatic animals, means there will be increased BOD for the aquatic life. Now next question, how are viroids different from the viruses? Okay, this question is from the NCERT section 2.6. You can find the answer here, but we will discuss the answer here. So viroids, you know how the viroids are different from the viruses. So viroids, they lack the protein coat. That's why they are smaller than the viruses. Then viroids consist of only free RNA, which is infectious, means it can cause infections, it can cause disease. Usually it infects the plants. And viroids, they consist of just free RNA, which is of low molecular weight. Viruses are nucleoproteins, means they consist of nucleic acid and the capsid also, which is known as the protein coat. So this protein coat, it protects the genetic material, which can be RNA or DNA. So these are the difference between virus and the viroids. Viroids are small infectious agents with single-stranded RNA without a protein coat. That's why they are smaller than viruses also. Viruses, they have single-stranded or double-stranded RNA, or they can have DNA also as the genetic material. And this RNA or DNA or the genetic material is present within the protein coat. This protein coat protects the genetic material of the virus. Viroids are very small in size compared to viruses. Viroids infects only plants, whereas viruses can infect plant, animals, and the other microorganisms like bacteria also. So viruses which infects bacteria, they are known as bacteriophage. Here. So here you can see this is the difference between viroids and viruses. Viroids are single-stranded RNA. Okay, viruses they can consist of DNA on RNA. This DNA can be linear or circular. Now in viroids protein code is absent. In viruses protein code is present. Protein code is also known as capsid. Viroids they are smaller, usually less than 300 nucleotides, and viruses they are larger as compared to viroids, and they can range from tens to 100 of nanometer. Now viroids they self-replicate in the host cells. Viruses they require host cell machinery for replication. So viral DNA it gets incorporated in the host genome. It hijacks the host machinery to produce more of its kind. Okay. Then viroids they cause diseases in the plant. Okay, especially they infect the plants. Viruses they cause disease in plants, animals, and humans. Then viroids they infect the plants. Viruses can infect plant, animals, bacteria, fungi, and others. And viroids, you can say, they cause disease like potato spindle tuber disease. Okay, clear. And this is the example of viroid, potato spindle tuber viroid. Viruses, there is influenza virus, HIV virus, and SARS-CoV-2 virus. Next question, describe the 4 major groups of protozoa briefly. So you can find the answer in the NCERT section 2.2. Okay, let's discuss the answer here. Okay, so first of all, first one is the amoeboid protozoa. So protozoans, they are divided into 4 groups. First one is amoeboid protozoans. They are also known as sarcodines. So they come under the group Sarcodina. Okay, so amoeboid protozoans, they are also known as sarcodines. They are characterized by the ability to move and feed using cytoplasmic extensions, which are known as pseudopodia or false feet. So using this pseudopodia, or what is the function of the pseudopodia? The pseudopodia helps in the movement of the bacteria, as well as the pseudopodia help in engulfing the food or prey as well. So they lack definite shape and often exhibit fluid and gelatinous appearance. Amoeboid protozoa are primarily free living, found in various aquatic and terrestrial environments. Now, some notable examples include Amoeba proteus and Antiamoeba histolytica. Antiamoeba histolytica is a parasite. It is a parasitic amoeba that causes amoebic dysentery in the human. So this is very important. You should remember this. Okay. You should remember about these parasitic amoeba which cause this disease. They can ask you questions in the NEET exam. Okay, I've already told you in the previous sessions that which topics are important for the NEET exam. But here we are discussing the NCERT exercise answers. Next one is ciliated protozoans. They belong to Coeliophora. They are also known as ciliates. Okay, they are characterized by the presence of numerous hair-like structures called cilia. So what are cilia? They are hair-like structures which cover their surface and are used for the locomotion as well as the feeding. So cilia, remember what is the function of the cilia? They are used for locomotion also, and they are used for feeding also. So how they are used for feeding cilia? So example of ciliated protozoan is paramecium. Parameciums have thousands of cilia on its surface. So due to the coordinated movement of the cilia, what happens? The water along with the food, it enters into the gullet of the paramecium. Okay, so this is how cilia helps in feeding also. Now, these organisms possess a complex cellular structure, including well-developed nucleus and organelles such as contractile vacuoles and food vacuoles. Now, ciliate exhibit diverse shapes and sizes, ranging from microscopic to visible, with the naked eye. Common examples include Paramecium caudatum, a widely studied freshwater ciliate, and Stentor roselli, known for its triumph-shaped and large size among ciliates. So you should remember the examples of all of these. Then flagellated protozoans, they belong to Mastigophora. So they are known as flagellates. They consist of the flagella, which helps in the locomotion. So they have whip-like structures known as flagella, which are the mode of locomotion. So they may be free living or parasitic. They are found in various aquatic and terrestrial habitats. Flagellates, they exhibit wide range of forms from single cell organisms to colonial forms. Examples is Trypanosoma brucei, which cause the African sleeping sickness, and Euglena also is a flagellated protozoan. Euglena, this is mixotrophic, means it can photosynthesize also in the presence of light, and it becomes heterotrophic in the absence of the light. So Euglena has both plant characteristic, which is it can photosynthesize, and it has animal characteristic also, means it becomes heterotrophic, or it follows heterotrophic mode of nutrition in the absence of the light. So Euglena shows, okay, that there is a common ancestor for both plants and animals. The next ones are sporozoans. Okay, they are also known as epicomplexans. So this is a little bit of, you can say, extra information here. They are non-motile parasites. Okay, characterized by the presence of a unique apical complex used for host cell invasion. So basically, sporozoans, why they are called sporozoans? Because they produce a stage which is spore-like. So their life cycle is very complex because they involve multiple hosts and distinct morphological forms. Let's take example of Plasmodium. Plasmodium causes malaria. Okay, so this Plasmodium has two hosts, human host and the female Anopheles mosquito. It has two hosts and it has distinct morphological forms. So when the Plasmodium enters into the humans through mosquito bite, it enters in the form of sporozoites, which is a infectious stage. Then the sporozoites, they travel through the blood, they reach the liver cells, which are known as hepatocytes. They will divide there by sexual reproduction. Then they will form merozoites, schizons, and then ultimately they will again infect the RBCs. Then again, in RBCs they will multiply. Then they finally form the gametocytes, which is a sexual stage. These gametocytes, again, it will enter into the female Anopheles mosquito. When the female Anopheles mosquito bites this person, then in the female Anopheles mosquitoes, what happens now? These male and the female gametocyte fertilize with each other, fuse with each other. So their fertilization takes place, leading to the formation of sporozoites. These sporozoites are stored in the salivary glands of the female Anopheles mosquito. Their life cycle is very complex. Example is Plasmodium falciparum, which causes malaria, Toxoplasma gondii, which causes toxoplasmosis, Cryptosporidium parvum, which is responsible for cryptosporidiosis. Now plants, they are autotrophic. Can you think some plant that are partially heterotrophic? Okay, let's discuss the answer. So usually plants are autotrophic, means they can photosynthesize, they can prepare their own food, they can convert light energy into the chemical energy, or they can convert light energy into food, you can say. Okay, clear? But there are some examples of the plant that show partial heterotrophy. Let's discuss them. The first one is the epiphytic plants. So epiphytic plants, such as certain species of orchids and bromeliads, they grow on other plants or structures but do not parasitize them. Okay, means they are not parasitic on them, although they are growing on them. These plants derive water and minerals from the air, rainwater, or decaying organic matter accumulating around them. While they may still perform photosynthesis, they rely partially or on external sources for the nutrients. Okay, these epiphytes, they grow on other plants, but they do not parasitize them. Means they are not obtaining nutrients from them, they are not harming them. They are obtaining nutrients from the either air or from the rainwater or from decaying organic matter. But they grow on the plants without harming them, without obtaining nutrients from them. Okay. Main one are these carnivorous plant. Okay, so this is very important. Carnivorous plant. Okay, these carnivorous plant supplement their nutrient uptake by capturing and digesting small insects. Okay, so basically they obtain nitrogen from these insects. So they can perform photosynthesis, but these carnivorous plants have evolved specialized structures such as modified leaves or traps to attract and capture and digest the prey. Okay, which are usually insects. Now examples are Venus flytrap known as Dionia muscipula, then pitcher plant, Nepenthes khasiana, sundews known as Drosera, and bladderwort known as Utricularia. You should remember this is very important. Question can come from here. These are all carnivorous plants. Now the question is, what do the term phycobiont and mycobiont signify? Okay. Let's discuss the answer. Phycobiont. The phyco term is related to algae. Now, phycobiont is the photosynthetic partner in the lichen symbiosis. So, you must be knowing about lichen. Lichen is a symbiotic association between the algae and the fungus. Okay. The algal partner is known as a phycobiont, and the fungal partner is known as the mycobiont. Now, how it is symbiosis? Both the partners are benefited. The phycobiont, which is the algae, it can photosynthesize and it provides the carbohydrates to the fungi, and fungi in return it provides the nutrients, minerals as well as water to the lichens. Also, this fungus gives shelter and protection to the algae because the algae is embedded in the matrix of the fungus. So basically, the body of the lichen, it is mostly made of the fungal part. Lichen is occupying, the algae is occupying little part. Most of the part is occupied by the fungus. And the fungus is providing shelter and protection to the algae. The algae is embedded in the matrix of the fungus. So this is a symbiotic relationship. Both the partners are benefited. The phycobiont, they are typically green algae or they can be cyanobacteria also. Alright, so I've already told you about the phycobiont and mycobiont. Mycobiont is a fungal partner in the lichen symbiosis. It provides protection to the lichen, also provides nutrients to the phycobiont. Okay, now mycobionts belong to the, you can say, Ascomycota or Basidiomycota, with Ascomycetes being the most common group associated with lichens. So you should remember this line. So if someone asks you the mycobiont belongs to which class, they belongs to Ascomycota or Basidiomycota, and especially Ascomycetes. Ascomycetes being the most common group associated with the lichens. Clear? Now next question, give a comparative account of classes of different of kingdom fungi under the following mode of nutrition and mode of reproduction. So we'll understand the different mode of nutrition and reproduction in different classes of the fungi. Let's talk about the mode of nutrition in Phycomycetes. So they are primarily saprophytic fungi obtaining nutrients from decomposing organic matter. Some species can be parasites also, invading and feeding on living organisms or other fungi. Then mode of nutrition. Let's talk about Ascomycetes. So they exhibit different modes of nutrition. They can be saprophytic, parasitic, or they can be symbiotic. Means they can form you can say association with algae where they form lichens, and they can also form mycorrhiza. Myco means fungus, rhiza means roots. So when the fungus is associated with the roots of the plants, it is known as mycorrhiza. Again, this is a symbiotic relationship. Some are pathogens also. They cause diseases such as powdery mildew and Dutch elm disease. So you should remember this. Now, mode of nutrition in Basidiomycetes. Basidiomycetes include wide range of nutrition strategies. So they can be saprophytic, parasitic, and as well as mycorrhiza. So they form ectomycorrhiza. Okay, Basidiomycetes, they are decomposers. They can break down complex organic matter. Then some are ectomycorrhiza, forming symbiotic association with the roots of the trees, while some are pathogens to plants and animals. Then let's talk about the Deuteromycetes, known as the fungi imperfecta. So Deuteromycetes, basically they are decomposers. Okay, so they lack, or the sexual phase is not discovered in the Deuteromycetes. Now they can be saprophytic. Mostly they are saprophytic. They are decomposers, but some can be parasitic, or they can form symbiotic relationship with other fungi such as Trichoderma. So these are the examples, Aspergillus and all. Okay, Candida species also. This can cause disease in humans. Now, mode of reproduction in Phycomycetes. So they can reproduce sexually and asexually. Asexual reproduction is through the formation of sporangia, in which spores are formed. In sexual reproduction, what happens? Zygospores are produced. Remember, so this is very unique feature. Remember, the zygospores are formed in the Phycomycetes. Zygospore is a, you can say, diploid structure. It is formed by the fusion of the two compatible hyphae. Okay, of opposing mating type, means plus and minus. When they fuse together, means plasmogamy will occur. Then there will be, you can say, karyogamy. So zygospores are formed in Phycomycetes. Then let's talk about the mode of reproduction in Ascomycetes. So they can reproduce. They have different strategies, reproductive strategies. So they can reproduce by sexual and asexual method. So they reproduce asexually by conidia, which are produced on conidiophores. Now the sexual spores are known as ascospores. So 8 ascospores are produced through meiosis, okay, which is followed by mitosis also. And these ascospores are produced in asi, which is sac-shaped. And let's talk about mode of reproduction in Basidiomycetes. So they can reproduce. So they reproduce, you can say, by sexual methods. If we talk about asexual reproduction, they only reproduce through fragmentation. So you remember this. In Basidiomycetes, there is no such asexual reproduction. They only, you can say, multiply by fragmentation. Clear? So asexual reproduction is not much prevalent in Basidiomycetes. Sexual reproduction involves formation of basidia, in which 4 basidiospores will be produced through meiosis. Now in Deuteromycetes, the sexual phase is not discovered. They only reproduce by asexual methods, by the production of the conidia. Sexual reproduction is absent or not well understood in this group. Now next question, what are characteristic feature of euglenoids? So this is very important question here. So euglenoids, what are the typical features? There is absence of the cell wall. Instead of the cell wall, pellicle is present. A protein-rich layer known as pellicle is present. So this is an animal feature, lack of the cell wall and present of the pellicle. Then they have true flagella of different lengths. They are autotrophic in the presence of light. This is a plant feature. And they are heterotrophic in the absence of light. This is the animal feature. So euglenoid, they have both animal-like features and plant-like features as well. So example is euglena. Okay, this is very important. They have both plant-like and animal-like features. Now next question, give a brief account of viruses with respect to the structure and nature of the genetic material. Also name 4 common viral diseases. Let's talk about the structure of the viruses. So they have the genetic material, which can be DNA or RNA. Now this genetic material, this genetic material carries the information necessary for viral replication and viral assembly, and only the genetic material can cause infection because genetic material gets incorporated into the cell. The capsid, it doesn't enter the host cell. Capsid remain outside, only the genetic material enters the host. Now they consist of a protein code known as a capsid. This capsid protects the genetic material. Capsid is composed of subunits known as capsomers, which are arranged in various symmetrical patterns. Now, sometimes in some viruses, envelope is present outside the capsid. Okay, so in some viruses, additional outer layer called an envelope is present. Now, this envelope may contain various viral glycoproteins. And what is the role of envelope? They play a role in viral attachment to the host and also help in the entry into the host cell. Okay, so basically the virus consists of the genetic material and the capsid. So in some viruses, the envelope is also present. The nature of the genetic material, so there can be DNA viruses. Okay, DNA can be double-stranded like in bacteriophage, or they can be single-stranded DNA as the genetic material. Examples you should remember: herpes viruses, adenoviruses, and papilloma viruses. You should remember the name of these viruses. Then you have RNA viruses, which have RNA as the genetic material. They are also known as the retrovirus. Okay. So RNA viruses may have single-stranded or double-stranded RNA as the genetic material. Usually the plant viruses, they are RNA viruses. Okay. Sometimes other viruses also, like HIV, it is also RNA virus. Hepatitis C virus, this is also RNA virus. Influenza virus, this is also an RNA virus. Then common viral diseases, influenza, which is known as flu. It is very contagious. It can spread from one person to another. So symptoms are fever, cold, sore throat, muscle ache, and fatigue. These influenza virus belong to the family Orthomyxoviridae. The next one is common cold. So this is infection of the upper respiratory tract caused by rhinovirus. Symptoms are again sneezing, runny or stuffy nose, sore throat, and cough. Then there is HIV virus, which causes AIDS. HIV is human immunodeficiency virus. So what happens in AIDS? The immune system is compromised because the HIV, it targets the CD4 T cells. The T cells, they are the immune cells. They provide immunity. So number of T cells, they decrease. So the immunity keeps on decreasing. Okay, so clear. So AIDS is known as acquired immunodeficiency syndrome. So slowly what happens during the lifetime, the immunity slowly decreases. That's why it's known as acquired immunodeficiency syndrome, and the person gets susceptible to opportunistic infections. HIV belongs to the family Retroviridae. It is a retrovirus. Then next, coronavirus in 2019 or COVID-19. This is also a retrovirus. Okay, so COVID-19 is caused by the coronavirus, which is SARS-CoV-2. Symptoms range from mild respiratory illness to severe pneumonia, acute respiratory distress syndrome, and multi-organ failure. It was a global pandemic in late 2019. Now question, organize a discussion in your class on the topic, are viruses living or non-living? Okay, so are they living or non-living? They are a connecting link between living and non-living. So what are the non-living characteristics? First of all, they are acellular. They don't have any cellular structure. They can be stored in bottles like crystals. So outside the host, they are inert crystalline structures which can be stored in the bottle. So viruses can be crystalline. Okay. They lack their own metabolism. They cannot produce energy, synthesize protein, or maintain respiratory and digestive symptoms. They cannot grow or multiply outside the host. Outside the host, they are considered non-living. They are inert crystalline structures. Now, what are the living characteristics? So they are host specific. They will infect a particular host. They have genetic material in them, which can be DNA or RNA. They can multiply inside the host. They have antigenic properties, means the immune response can generate against the virus. So the immune response is generated against the antigens. So virus, they have antigenic properties. Immune system is activated against these viruses. They are obligate parasites, means they require a living host for their growth and multiplication. And in viruses, mutations also occur, which is again a feature of the living organisms. So this was the end of this session. I hope this session was quite knowledgeable for you. Thank you very much for listening.